Okay, great. So thank you, everybody, and sorry for changing to English. I have zero knowledge of Russian, but I hope that this is fine with you. So what we want to present today is a project that we built around the innate lymphoid cells. And we chose this particular cell type and to try to dissect the heterogeneity because it's also a very challenging cell type. And we know that we have been hearing a lot of talks around single cell RNA-seq, but sometimes you require much more than just the single cell RNA-seq to do your experiments. And we think that innate lymphoid cells, for example, is one example that you can combine different platforms that can create more power to dissect your research. So innate lymphoid cells are a growing family of immune cells, and they mirror the phenotypes and functions of T cells. You can see here natural killer cells can be also considered the innate counterparts of the cytotoxic CD8 T cells, whereas ILC1, ILC2, and ILC3 they may represent the innate counterparts of these CD4 helper cells. However, in contrast to T cells, ILCs do not express antigen receptors or undergo clonal selection, either expansion when they are stimulated. Instead, they react promptly to signals from infected or injured tissue, and they can produce an array of secreted proteins, for example, cytokines. And therefore, they direct the developing immune response into that is adapted to the original insult. Thus, the power of ILCs may be controlled or unleashed to regulate enhanced immune responses. And this, of course, can be useful to do disease prevention or therapies. But if we focus on the ILCs specifically, they also have been shown to be highly heterogeneous. And a comprehensive phenotyping is still elusive. For example, some years ago, there was a report that showed that ILC1 population had a lot of contaminated population of cells that were expressing CD4. This was later acknowledged that it was indeed a subset from the ILC ones. So you can see that there is still not a lot of concordance. So there is still a lot of knowledge that we need to refine that characterization. But also the cells, they accompany with several challenges. So they are very difficult to enrich. And nowadays, if you would just work with a PBMC population or a whole blood population, you would have a lot of cell types that you would not use the information for. And that's a lot of money that you're paying for sequencing for nothing. Therefore, it is important to find ways of how to enrich these cell populations and how to combine different platforms that can help you to achieve that. And we came up with this complete workflow to analyze, for example, this challenging population. And this is an example, ILCs, but this can be, of course, any other cell type that you could imagine that it's hard to get. So what we did is we have our samples and we combined fluorescent conjugated antibodies plus oligoconjugated antibodies, both of them to stain our cells. Then we sorted our cells based on lineage markers to focus on the total ILC population. And once we had the enriched population, we submitted it to the BD Rhapsody workflow to achieve the single cell resolution. We use not the whole transcriptome approach. We use a targeted approach because we wanted to focus in a more effective way, so just a few genes. And we're talking about 399 genes and 42 oligoconjugated antibodies that could be assessed by sequencing. Once you get all of the results from that, what we did, we just submitted it to our bioinformatic pipeline. And the knowledge that we obtained from this data set was actually used to perform a flow cytometry panel that could help us to interrogate with more throughput and more number of the patients and on a daily basis approach into an analyzer. And I wish I can show you along all of the presentation how this can also serve to validate your single cell RNA-seq data, but also can get you closer to have a more impact into, let's say, the translational space. And of course, because we are combining different technologies, we would like to see them that they are concordant. So we took a aliquot of the original samples, and we first enumerate by flow cytometry what was the proportion of cells that we're having per donor. This can also help you to, that if you're combining, for example, a flow cytometer and a single cell RNA-seq experiment, if you have some knowledge that you can input into the cytometer, can help you to stratify samples or stratify patients that would make sense to put in the single cell RNA-seq pipeline. Because, I mean, it is more expensive to do a single cell RNA-seq experiment. So we took an aliquot just to visualize that the sample that we have has the 3 ILC populations, because this also varies a lot between donors. And then we decided to do a sorting strategy where we could filter out our ILC population, so the total ILC, and you can see here that by lineage exclusion and the negative expression of CD56, we chose the ILC populations. It is very hard to do a proper gating in here. So you might have the challenge after that you don't have a very pure population. But the flow cytometer also has a limit of markers that you can use. And therefore, you cannot put whatever protein that can help you to resolve this possible contaminations, or to be very clear that you're working with the ILCs. The good thing is that, for example, if you use the Rhapsody system, you could combine this oligoconjugated antibodies on top of the genes. And this can also help you to exclude the contaminants that you have. And you can do what is so-called a post-sorting purity check of your cells. That sometimes is very hard when you have very rare populations, like, for example, the ILCs. If you want to do a downstream analysis, it is very hard to sacrifice those few cells to do this post-sorting purity check. And we included some markers like CD19, CD14 for B cells. ILCs are excluded for the expression of CD3, and then also the expression of CD94. So we could, using the Rhapsody data, the Rhapsody obtained data with this additional protein markers, oligoconjugated antibodies, we could gate our in our analysis to focus in the 3 ILC populations that were later defined by the expression of CD117 positive ones. Those would be the ILC3s, the positives for CD294, ILC2, and ILC1 as the double negative for both. So we have now our pure population for the analysis. And of course, as I mentioned, we enumerate, or we first check an aliquot of the samples if we would have sufficient ILCs to work with. And you would like to maintain the same frequency when you're working with 2 different platforms. And this is super important because you need to choose a system that you can validate in other platforms and that respects or doesn't have or create any bias in your cells. And you can see here that the 3 donors that we took and that we use for the Rhapsody analysis, the cross-platform validation was very concordant. So we still have the same percentage, even though that on the Rhapsody and in any single cell RNA-seq platform, you can never achieve the throughput of the flow cytometry. But nevertheless, the percentages were very concordant. So then we were very confident in our data. And of course, If you do this manual gating with a lot of parameters, you would end up being lost. So what we did is, based on those 399 genes and 42 oligonucleotide proteins, antibodies, we selected those highly dispersed ones, and we did just a t-SNE, and we could identify 3 major groups that actually, when you overlay the manual gating, they very nicely represent the 3 ILC populations. But of course, we had the protein information on top of that, so we could explore in more detail the distinct group of cells and how these markers were expressed. And you can see here in this part of the t-SNE, this is representative of the ILC1 population. And you can see that, as I was mentioning a few slides ago, there is a population of CD4 positive cells. There is a bit of a controversy of if these positive CD4 cells are exclusive for the ILCs, and you can see that indeed we can confirm that they are exclusive on these ILCs, but there are also a population of those that are not. So this also reveals heterogeneity within the ILC population. And that is not only applying to ILC1, so you can see for ILC2, for example, you have markers like CD27 that they are not expressed with the same uniformity along of the cluster. That can also be applied for the ILC3 population, where you can have, for example, CD2 that is, of course, not homogeneously expressed, and that entails heterogeneity within. So then you could focus in this particular clusters and analyze more deeper. So we took those 3 manually gated populations and we did a differential gene expression analysis. And we were very happy to see that using that combined manual gating and the unsupervised way by doing this differential gene expression analysis, we could identify some transcription markers that are being reported to define these populations. For example, TVX21 for the ILC1, it is differentially expressed in this population, or GATA3, for example, for ILC2. It is upregulated in this population, or ROR-R for the ILC3. So this creates, of course, a lot of power, because you have also additional markers that you can help to identify your cell populations. But of course, the benefit of having in the same experiment both protein and gene expression, this allows you also to plot correlations between gene and protein, and also can help you to identify markers that are relevant either at the protein level or at the gene level, or maybe they are concordant and you can use them at both. And just to focus on the ILC1, you can see that CD4, it is exclusively expressed not only at the protein level, but also at the gene level. And we just look a little bit on the literature, and indeed this observation was in the agreement of some reports that show that only the ILC1 population shows an heterogeneous population that expresses CD4, but not only CD4, also CD8. And you could dissect this population nicely. So what we wanted to do next is to focus and to further confirm that in our plot, in our data, we had this distinct population within the ILC ones. So we took the 3 ILCs manually gated and we plotted against CD4 and CD8, and you can nicely see that you can resolve a population that is expressing CD4 or CD8 or the double negative one. We could manually gate on those populations, and then you can do a differential gene expression analysis. And this is just a heat map that displaying this representative protein and gene signatures associated to each subset. And you can see that this illustrates very nicely the high heterogeneity and the complexity of the ILC subsets. And this is not only genes, this is also proteins. So you could potentially discover within here some markers that can help you to, for example, design a flow cytometry panel to dissect these three populations within. So this is what we did to deeply characterize them. And of course, in here, you can put a limited number of donors, but imagine that you want to go into a larger cohort where you have hundreds of donors and you want to do them on a daily basis. And if you think about single cell RNA-seq, we are still not there. It would be very expensive. In this case, we have identified by having protein and gene expression markers, we configured a panel that could inform better inform how we can do this panel to gate, for example, in these three different populations, and then explore a larger number of donors. And you can see that we have some markers that are oligoconjugated proteins that we put, so we can directly configure the panel just by choosing the appropriate fluorochrome. But there are some other markers, and I want to draw you the attention to, for example, CD63. CD63 is a marker that we identify here to be positive for the double negative population. And this is not a protein, this is just a gene. But because we have plotted also some correlations, we decided to use CD63 also as a protein to see if we could further dissect and use this marker. And it is a gene expression marker. And if we expect to have a correlation, then it might be that in the flow cytometry and at the protein level, we could see this marker to be different. So we just grabbed several donors. First, we use the same donor to try to validate our data. And so we follow the gating strategy to focus on the ILCs. And for this, we use a Symphony analyzer, which is a high parameter flow cytometry. So you could include those 12 markers plus additional ones. And we took the ILC1 population by our gating, lineage gating. And then we further dissect this population based on CD8 and CD4 expression. And then you can see that these additional markers that we found from the RNA-seq data, they also show consistent expression and also differential expression. And you can see that CD63 also, nevertheless, we found it as a gene marker. We decided to include it in the panel as a protein marker. You can see that there are some differences between the ILC1 population as it was expected. But of course, also in high parameter flow cytometry, it's very hard to...
I'm sorry.
Yeah. Okay. Yeah, it's just the last slide, last two slides, but I will just illustrate that we have also some plugins that can help you to dissect each of the clusters and to identify what are the markers that are defining this cluster. And there you can even do a validation of this unsupervised clustering by doing a manual gating strategy. And you can see that very nicely you can separate your clusters in flow cytometry data. And this, of course, can help you to analyze across different donors and in a larger population. Yeah, and this is again just to illustrate that the power that you can have combining different platforms, and that can help you to explore much deeper your rare populations. So thank you, and if you have any question, please let me know.